Hello, and thank you, everyone. So I'm going to tell you about how I view human biology in a very different way through the lens of data and of machine learning. For most of my career, I've told people that I'm bilingual, and they probably think that I'm referring to the fact that I grew up speaking Hebrew and then moved to speaking English, but that's not really what I mean. My first language as a researcher is actually the language of machine learning. I've been doing machine learning since the early 1990s, long before this latest wave that we are all familiar with. So that really is my first language. But over the course of my career, I became increasingly drawn not just to the beautiful mathematical abstractions that machine learning can offer, but also to how it can help transform the world. Andrew spoke about a number of different applications that machine learning has been applied to, some of them less inspiring, some more so, but one that I've always felt drawn to because of its tremendous impact on all of us, on all of humanity, is the field of biology and human health. So to me, my second language, which I've been doing since the late 1990s, is the field of biology and how machine learning can be applied there. Machine learning, my first language, as Andrew, I think, correctly described, has really been transformed in the last few years in tackling an array of problems that really would not have been imaginable even a few years ago. So if you'd asked me even 5 years ago about the feasibility of machine learning as applied to image recognition at the level of human performance, I would have said, that's an amazing task, and maybe in a decade or two we might get there. But that's actually not where we are. So if we look at this image over here, in 2005, we might have been able to ask the computer, is there a bear in this image? And for this dichotomous task, the computer might have been able to give you the answer maybe 5 to 10% of the time. Fast forward less than a decade, you would be able to show this image to a computer, and it would say, I think there's a wolf here, which is not quite right, but at least is directionally going in the right direction. Fast forward 3 years, notice the shrinking of the time gap. The label that a computer did in fact apply to this image is a brown bear swimming in the water. Now, again, it's not quite right, but certainly it's a reasonable description that is sort of almost correct. 2 years later, this is the actual caption for this image. So at this point, if you actually look at quantitative data, machines are able to caption images at beyond human level performance. And this is for images that people are actually good at looking at, like this one. For images that people are not so good at looking at, like images, for instance, of cells or of tumors, which is not our natural domain, computers far exceed human level performance in interpreting and finding signals in those images. The key drivers to this success have been threefold, and these will figure in some of my later discussion. Most important, perhaps, is the existence of massive data. And if you look at the places where machine learning has been most successfully applied, areas like image recognition, speech understanding, natural language translation, those are places where natural data is abundant and can be used for training the kinds of models that we'd like to build. A second component in the success is the availability of incredibly powerful computational resources, as demonstrated by powerful processors, large amounts of memory, and of course the cloud that makes this available at almost like turning on the water tap. And those together have enabled the creation of much better models that are able to leverage this data and this compute power to make more accurate predictions. And it turns out that when you put all these together, big data really do help drive a huge performance increase in terms of these models. So this is a picture that's adapted from a cartoon that Jeff Dean, who is the leader of Google Brain today, typically shows, which is that simple models, it really doesn't much matter how much data you give them. At some point, they asymptote at a certain level of performance, which is not that great. Whereas the kinds of rich models that we're able to construct today just keep going and going, and the performance keeps getting better and better. The reason for that is that the old models relied on people to define what is most salient in a large number of features. So given an image, a person would have to say, I think corners matter, or this type of textures matter. And it turns out that people are not really good at identifying what is important signal in complicated high-dimensional spaces. Today, we're able to train models that have multiple layers of abstraction, and they are able to discern for themselves what are the important features that emerge out of the data and construct the signal that is most informative about the task that we care about. So given an image such as that of a wolf, initially the computer will automatically figure out what kinds of edges and textures are important, put those together to define corners and larger level elements like the eye of the wolf or the ear of the wolf, and together at the very end there will be the ability to create the category of wolf versus non-wolf. But all those intermediate layers of representation are constructed automatically by the computer via end-to-end training. That's important in images, and it's even more important in areas like biology and health, where human understanding is intrinsically more limited because it's a domain that we just don't really understand yet. So this type of structure is something that now provides us with a new lens for looking at the data. So if you look at a random image database and you put it through this kind of levels of processing that I just described, and then you kind of visualize the result, it turns out that the computer automatically discovers in this many hundreds of thousands of dimensions of the space a lower dimensional manifold, a substructure that allows images that, although from a raw pixel perspective might appear quite distinct from each other, but are semantically related, it will place them next to each other in this lower dimensional manifold that spans this very high dimensional space. So images of cars, even cars of different colors with different poses, will appear next to other cars, and images of cats will appear next to other cats, and also adjacent to images of dogs, even though they're different categories, because semantically they are similar to each other. And this structure is discovered completely autonomously by the computer without the need for a person to tell the computer that cats are kind of similar to dogs. Now, it turns out that this lower dimensional space captures fundamental meanings, again, that you would not necessarily expect a computer to be able to figure out. So, for instance, in some recent papers, they've shown that the computer figures out simply by looking at the semantic context, the syntactic context, the word context by which a word appears, that king is to queen as man is to woman, or that walking is to walked as swimming is to swam. Or that Russia is to Moscow as Japan is to Tokyo. And all of this just emerges naturally from the patterns that the computer is able to discern in very large amounts of data. Now, this is one of my favorite examples because it allows you to sort of really do something that almost seems like magic. So the computer is able to take images of a man with glasses, multiple images of men with glasses, subtract in the semantic space images of men without glasses, add in images of women without glasses, and generate images, plausible images of women with glasses. Now, if you try to do that in raw pixel space, you get gobbledygook. So it's really because we have this underlying semantic representation that the computer is able to do that. So that's my first language, where you can see that a lot of advancements have come. And in parallel, I'm now going to talk a little bit about the advancements of my second language, which is biology and medicine. So here, too, we've seen in the last 50 to 70 years huge advancements emerge. In the 1950s, the polio vaccine. In the 1990s, biologic therapeutics that, for instance, provide anti-inflammatories that are currently helping millions of people with autoimmune diseases. In the 2010s, cancer immunotherapy that is providing cures to cancers like melanoma that were a death sentence as recently as 5 years ago. 2012, cystic fibrosis, where people who were dying at the age of 20 are now able to live long and healthy lives because we have understood the specific genetic lesion that drives their particular version of cystic fibrosis and provide a targeted cure based on their genetics. And today, just in the last year, cell therapies where active engineered cells were put into a person's body and like heat-seeking missiles kill the tumors that we're about to kill them. So really, an incredible amount of progress in the last 70 years. But unfortunately, as Young eloquently told us, that progress is not really heading in the right direction from a financial perspective. Whereas in the 1970s, it cost $100 million to identify a new drug. Today, it costs $2.5 billion. Now, why is that? I mean, we have all these improvements in efficiency, better assays, better experimental methods. Why is that? It's because the funnel by which drug discovery is done is becoming increasingly less efficient. So today, if we look at the number of compounds that are put into drug discovery at the very beginning of the funnel, which is maybe 10,000 compounds, when you get to preclinical development, it's 250. You get to clinical trials, it's 5, and when you get to a regulatory approval, it's 1. So that $2.5 billion is not because the process for a single compound has become less efficient, it's because we have so many more failures and less ability to predict at the beginning what's going to work and what's not. So in some sense, what we're faced with is a challenge in building predictive models. And so if you put those two together, this quote from Thomas Friedman, who actually wrote this quote, I love this quote, and Thomas wrote this about Coursera, which Andrew and I co-founded, which is that big breakthroughs is what happen when what is suddenly possible meets what is desperately necessary. Suddenly possible is the ability of machine learning to make really good predictions about things that are unknown at the start. And desperately necessary is this crisis in drug development and the fact that we are going to be spending $18 trillion on healthcare if we don't stop this trend. So how do we see those two things fitting together? I told you at the beginning that machine learning and the successes of machine learning rely on the availability of big data. 7 years ago, when Andrew and I founded Coursera and I left Stanford, big data in biology was a couple hundred samples. But that world has changed. So if you look at this graph of the number of human genomes sequenced over time, starting from the very first human genome back in 2000 and going until today, and note that this graph, the y-axis, is on a log scale, you see that there's this clearly increasing linear trend, which corresponds, of course, to an exponential growth in the number of human genomes sequenced. And what's really interesting is that that growth is twice as fast as Moore's law, if you look at that slope. So we are currently in a position that, depending on whether you think this trend will continue or slow down to be only as fast as Moore's law, will be somewhere between 100 million to 2 billion genomes sequenced by 2025. That's a lot of data. We're also starting to put that data together with some phenotypic data, like disease data, to try and understand how the different variants that we see in different people's genomes drive human disease. So again, if you look back to 2008, going forward up to 2016, every year we understand more and more variants that contribute in ways that, unfortunately, we don't fully understand to human disease. But at least we can identify them, which gives us the starting point to deconvolute the biology and hopefully give rise to treatments like cystic fibrosis. Now, in order to enhance that process further, various governments are creating big biobanks that serve the needs of scientific discovery in biology. So we can see that in the UK Biobank, one of the largest and best curated of those, there's data on 500,000 people with deep, deep phenotyping that includes not only their genetics, but also biomarkers in blood and urine, full body scans, brain scans, cognitive testing, activity monitoring, thousands and thousands of phenotypes, which give us the ability to now start mapping the genetics, those A's, C's, G's, and T's in our body, to the phenotypes that we see clinically. So clearly, this looks like a machine learning problem, right? You have the genetics, maybe the environmental factors on the one hand, you have the phenotypes that you're looking to predict on the other. We can just put those together and solve this. That's all very well and good until you start doing the math on this. And you realize that even in a population cohort that's been gathered recently, there's 261 million individual DNA variants that are detected at the population level. And this is just a subset of population that is primarily of European origin. If you look more broadly, it's going to be a much larger set than that. And of course, the phenotypic space is also incredibly high dimensional. So even with large cohorts like 500,000, or even if we were to go to a few million, you're never going to have enough data to solve this machine learning problem directly of mapping from genotype to phenotype. So this is where one of the ideas that we're trying to pursue at our work at insitro is to create a much larger data set that we can basically engineer by putting together biological technologies that allow us to provide an additional repository of data that will allow us to interpret and enhance this mapping. And we can do that because really smart biologists and biological engineers over the last few years have created huge advancements in biological data generation across multiple fronts. They have enabled the generation of cellular systems that are actually quite relevant to human disease. As recently as 5 years ago, most biological experimentation on cells was done on cancer cell lines, some of whom were harvested from people like the HeLa cells from Henrietta Lacks over 50 years ago, and have been passaged so many times that honestly they bear almost no resemblance to human biology. Today, using the Yamanaka factors, we're able to take stem cells and differentiate them into many, many different lineages that much better resemble real human biology. And it's not just individual cells, it's organoids and even multi-organ systems on chips that allow us to replicate reasonable pieces of human biology in a very scalable way. We can perturb those systems using very fine-grained molecular scissors, using technologies such as CRISPR, which allow us to turn genes on and off, to make edits at the level of individual base pairs so that we can now start to see what happens if you change this A to a C. What will it do to the cell? And we can measure those cellular systems in many, many different modalities, using advancements in optics, in mass spectrometry, in sequencing. So you can really start to get hundreds of thousands of different readouts on each one of those cellular systems. So you put these three together, and it's a perfect storm of data production if you just create the right environment. So that, in fact, is what we're building. We're building a factory for data, not just a factory for something else where you collect data as a byproduct, but rather a factory whose primary purpose is to create data that will drive the generation of machine learning models. And those machine learning models will, in turn, enable us to make predictions that are relevant to human biology, human disease, and drug development. So to me, if I take a big step back, I think the era that we're about to enter is a really interesting epoch if you think about the progression of science. If you look at science, it sort of progresses in relatively discrete epochs that sort of trend into each other. So the late 1800s were the epoch of chemistry, where we really started to understand the periodic table and how atoms contributes to how different substances interact with each other. That trended in the early 1900s to the era of physics, where we split the atom and we created fission and fusion. And that really sort of laid the foundation for modern transistors and silicon chips and so on, which gave us, in the 1950s, what began as the era of computing, which gave us what I don't need to tell you, everything that many of you are doing here today. That, in turn, created in the 1990s actually two parallel threads that up until now haven't really interacted. So the era of data, which is a direct product of computing and the ability to gather and store data at massive scales, and in parallel, leveraging on some of the advancements in chemistry and physics, but also in miniaturization and in computing, the era of biology, in which the human genome was sequenced, and all of these abilities to capture biological data at scale that drives biological experimentation were in fact enabled. The era that I see emerging upon us today is the era that actually synthesizes those two into a single unified discipline, in which biology meets technology, meets data into a single framework. And that framework in which biological data is collected using these incredible technologies that have been developed and interpreted using machine learning algorithms, which in turn define and induce new experiments in a closed-loop system, that is what's going to allow us to accelerate that process of drug development and other types of therapeutic modalities that will enable us to cure more people faster, cheaper, and better. And so, in some sense, This is the AI for good that I'm hoping to help enable in this next epoch. Thank you very much.